Real-world data analysis in clinical trials begins with a distinction that is easy to blur. Real-world data is information collected in routine healthcare settings outside the controlled structure of a trial. The important point is that value does not come from having more data, it comes from using fit-for-purpose data to support a clear decision. That matters because trials are designed to be focused and controlled. They usually involve selected populations and defined follow-up windows. Real-world data can extend that picture across broader groups of patients, longer time periods, and everyday patterns of care. It can help teams understand who is treated in practice, how care pathways work outside protocol conditions, and what happens once formal trial follow-up ends. The main sources include electronic health records, insurance claims, disease registries, lab data, and patient-generated data such as wearables or surveys. Each has trade-offs. Claims data is often structured and consistent, but it was built for billing rather than clinical interpretation. Electronic health records can be richer clinically and more longitudinal, but they are often harder to standardize across providers and systems. Registries can be especially useful in focused settings such as rare disease, though they may take time to build enough depth or scale. Teams often combine sources to improve coverage, but that adds work around linkage, harmonization, and governance. In practice, real-world data is usually complementary to trial data rather than a replacement for it. Before a study starts, it can support feasibility work, help define target populations, and show where eligible patients may be found. During development, it can help explain why outcomes seen in routine care may differ from outcomes seen under trial conditions. After a trial ends, it can extend safety and outcome monitoring over longer periods and wider populations, where rarer events or longer-term patterns may become easier to detect. The analysis itself is often harder than expected because patient records are fragmented. Diagnoses, procedures, prescriptions, and visits may sit in different tables or systems, so analysts have to reconstruct a coherent timeline from separate pieces. Some problems are straightforward, such as invalid dates or coding errors. Others are less clear. Prescription records may overlap because patients refill early, change dose, or stop and restart treatment. Visit records may not map neatly to a single healthcare encounter. Those decisions can affect the results, so cohort rules, exposure definitions, endpoints, and edge cases need to be defined early, applied consistently, and documented clearly. Where clinical judgment is needed, it should be turned into explicit traceable rules rather than left implicit. There are also important limitations. Data quality can vary, some populations may be underrepresented, and confounding remains a central challenge because routine care is not randomized. Privacy and governance matter too, even when data has been de-identified. So the practical starting point is simple. Define the decision first, choose sources that are fit for purpose, and avoid treating observational findings as if they provide randomized proof. To learn how Quanticate can support your next trial, email inquiries@quanticate.com or request a consultation through our website. Thanks for watching, and don't forget to subscribe for more practical insights on clinical trials and modern drug development.